Welcome to this rigorous scientific lecture on validating CRISPR editing performance. Today we're going to walk through a complete experimental workflow that takes you from designing your edit to validating it in new cell lines. This is not about assumptions. It's about measurement, quantification, and reproducibility. Let me start with a critical question. How do we know our CRISPR edit actually worked? Many researchers assume that if they design a guide RNA and transfect it into cells, the desired edit happens efficiently. But this is scientific malpractice. We must measure, validate, and quantify every step. Step 1 is defining your intended edit with absolute precision. Let's say we want to knock out the CCR5 gene to confer HIV resistance. We need to specify exactly what genomic change we're making. We target CCR5 on chromosome 3, specifically exon 4. Our guide RNA binds to this sequence, and Cas9 cuts 3 base pairs upstream of the PAM sequence. We're aiming for a frameshift mutation through non-homologous end joining. We must document the exact guide sequence, the PAM motif location, and the expected molecular outcome. Will it be a deletion, insertion, or substitution? What size indel do you predict? Document both the wild-type sequence and your predicted mutant sequence. This becomes your hypothesis that you will test rigorously. Step 2 is measuring on-target efficiency. This is where most researchers get it wrong. You cannot assume your edit worked. You must sequence the target locus and quantify the editing efficiency. Here's why sequencing is absolutely essential. CRISPR editing efficiency varies wildly, from 0% to 90% depending on the guide, cell type, and delivery method. Without sequencing, you're flying blind. The workflow is straightforward. Transfect your cells with the CRISPR components. Wait 48 to 72 hours. Extract genomic DNA. PCR amplify the target region. Then perform next-generation sequencing to analyze thousands of individual reads. The key metric is indel frequency. What percentage of sequencing reads show insertions or deletions at your intended cut site? This is your on-target editing efficiency. Let's do a back-of-the-envelope calculation. Say you sequence 10,000 reads from your amplicon. You have 10,000 total reads from your sequencing run. 4. 200 reads show the wild-type sequence with no modifications. 5. 800 reads show insertions or deletions at the target site. Your editing efficiency is 800 divided by 10,000, which equals 58%. This is a decent efficiency. But notice that 42% of your cells remain unedited. But we need to dig deeper. Not all indels are equal. Let's break down the types. Here's a realistic breakdown. Only frameshift mutations will knock out the gene. In-frame deletions of 3 base pairs or multiples of the third of May leave the protein partially functional. So your functional knockout rate is actually lower than your total indel rate. The functional knockout rate is 12% plus 9% plus 18%, which equals 39%. This is significantly lower than the raw 58% indel rate. This is why you must always analyze the functional consequence of your edits, not just the presence of indels. Step 3 is measuring off-target sites. This is arguably the most important validation step, because off-target editing can invalidate your entire experiment. Let me ask you a question. Why do off-targets matter so much? Because if your guide RNA cuts at unintended genomic locations, you're creating confounding mutations that could affect your phenotype. Your results become uninterpretable. Imagine you knock out CCR5 to test HIV resistance, but your guide also cuts a tumor suppressor gene. Any phenotype you observe could be due to the off-target, not your intended edit. Your guide RNA can tolerate 2 to 3 mismatches and still cut at off-target sites. These could be scattered across multiple chromosomes. You need to identify and quantify editing at each potential off-target. There are several methods for detecting off-targets. The gold standard is guide-seq or circle-seq, which provide unbiased genome-wide detection. A more practical approach for most labs is computational prediction followed by targeted sequencing. Use tools like CASO-F-Finder to predict the top 20 to 50 off-target sites based on sequence similarity. Let's walk through an example. You predict 30 potential off-target sites with 2 to 4 mismatches from your guide sequence. You have 30 predicted off-target sites to check. Design primers to amplify each site. Perform deep sequencing on all 30 amplicons. This costs about $500 for a multiplexed run. Here's what you might find. On-target editing is 58%, but off-target 1 on chromosome 7 with 2 mismatches shows 8.2% editing. This is significant and concerning. Off-target 2 has 1.1%. The remaining sites show less than 0.1% editing, which is near background. Now you face a decision. Is 8.2% editing at off-target 1 acceptable? That depends on what gene it hits. If it's in a non-coding region, maybe acceptable. If it's in a critical gene, you need a better guide. This brings us to step 4, comparing multiple guide designs. You should never rely on a single guide. Design and test at least 3 to 5 guides targeting the same gene. Why test multiple guides? First, it gives you options if one has high off-target activity. Second, consistent phenotypes across multiple guides provide strong evidence that effects are on target. Here's a comparison of 5 guides targeting CCR5. Guide D has the highest on-target efficiency at 81%, but it also has 12.5% off-target editing. Guide C has the best specificity with only 0.3% off-target, but lower on-target at 45%. Guide B offers a good balance, 72% on-target, with only 2.1% off-target. We can calculate a specificity score as the ratio of on-target to off-target editing. Specificity equals on-target percentage divided by highest off-target percentage. For guide B, that's 72 divided by 2.1 equals 34.3. Higher is better. So which guide should we choose? Guide C has the best specificity at 150, but its on-target rate of 45% might be too low for some applications. The answer depends on your application. For therapeutic applications where safety is paramount, choose Guide C or B. For research where you need high knockout efficiency, Guide B or D might be better, with appropriate controls. Step 5 is building a predictive model. Once you've tested multiple guides, you can start to understand what sequence features predict good performance. Modern CRISPR research uses machine learning to predict guide efficiency. But you can build a simple model, even with limited data. Here are features that correlate with efficiency. First, GC content. Optimal is 40 to 60%. Too low or too high reduces efficiency. Second, position-specific nucleotides. Guanine at position 20 immediately before the PAM improves efficiency. Third, secondary structure. Guides with strong hairpins or self-complementarity perform poorly. Fourth, chromatin accessibility. Guides targeting open chromatin regions work better than those in heterochromatin. A simple linear model might look like this. Efficiency is approximately a weighted sum of GC content, the nucleotide at position 20, and chromatin accessibility. Train this model on your tested guides. Even with just 5 data points, you can identify trends. With 20 to 50 guides across multiple genes, you can build a reasonably predictive model. Let's build a toy model with our CCR5 guides. Looking at this data, we see that guides with G at position 20 and high accessibility tend to perform best. Guide D has the highest GC content at 65% and the highest efficiency, but remember, it also had high off-targets. From this small dataset, we observe that having G at position 20 correlates with about 15% higher efficiency compared to other nucleotides. Now you can use this model to screen hundreds of potential guides computationally before testing. This saves enormous time and resources. The final step is validation in a new cell line. This is absolutely critical for establishing that your findings are generalizable. Why does validation in a new cell line matter? Because CRISPR efficiency is highly cell type dependent. What works in HEK 293 cells might fail in primary T cells. Cell type specific factors include differences in DNA repair machinery, chromatin state, Cas9 expression levels, and transfection efficiency. Take your best performing guide from HEK 293 T cells, which gave 72% efficiency. Now test it in JERCAT T cells. Will it maintain similar efficiency? Let's walk through a validation experiment. You take guide B, which performed best in NHEJ 293 T cells. Transfect into GERCAT cells using the same protocol. Extract DNA at 72 hours. Amplify and sequence the target locus. You find the efficiency in GERCAT cells is 54%. This is lower than the 72% in NHEJ 293T, but still quite good. Critically also check off-targets in the new cell line. Off-target patterns can differ between cell types due to chromatin differences. The top off-target in GERCAT cells is 3.1%, slightly higher than the 2.1% in NHEJ 293T, but still acceptable. But we're not done yet. The ultimate validation is measuring the functional phenotype. For CCR5 knockout, the functional validation is testing HIV resistance. Expose edited and unedited GERCAT cells to HIV virus and measure infection rates. Unedited GERCAT cells show 89% HIV infection. CCR5 knockout cells show only 12% infection. That's an 87% reduction, confirming functional knockout. This functional validation confirms that the on-target editing, not off-target effects, cause the phenotype. Let's talk about the complete timeline and costs for this rigorous validation workflow. Here's a realistic timeline. Guide design takes about 1 week. Initial testing of 5 guides takes 2 weeks. On-target sequencing adds 1 week. Off-target sequencing takes 2 weeks. Validation in a new cell line takes 2 weeks. Functional assays take another 2 weeks. Total time is about 10 weeks. Total cost is around $300, which is very reasonable for rigorous validation. This is the bare minimum for publication quality work. Cutting corners at any step compromises your conclusions. Let me highlight common mistakes that researchers make in CRISPR validation. Mistake 1. Testing only one guide. Always test multiple guides. If your phenotype only appears with one guide, it might be due to off-targets. Mistake 2. Skipping off-target analysis entirely. Some researchers assume modern guides are specific enough. They are not. Always check off-targets. Mistake 3. Using restriction enzyme digest instead of sequencing to detect edits. This only tells you IF editing occurred, not the efficiency or the exact sequences generated. Mistake 4. Only testing in easy-to-transfect cell lines like NHEJ 293T. You must validate in your biologically relevant cell type. Mistake 5. Assuming that any Indel equals a functional knockout. Remember our analysis showing that in-frame deletions may not disrupt function. Let's discuss important statistical considerations for CRISPR experiments. You need biological replicates, not just technical replicates. Perform the entire experiment at least 3 independent times. For sequencing, aim for at least 1,000-fold coverage of each amplicon. This gives you 0.1% sensitivity for detecting rare off-target events. With 1000x coverage, if you require at least 10 reads supporting an indel, your detection limit is 10 divided by 1000, which equals 1%. For 0.1% sensitivity, you need 10,000x coverage. This is important for rare off-target detection. Always include proper controls. Untransfected cells as a negative control. A non-targeting guide RNA as a specificity control. A positive control guide with known performance. Let me briefly mention advanced CRISPR technologies. Base editors and prime editors have different validation requirements. Base editors convert C to T or A to G without double-strand breaks. You must check for bystander editing at nearby cytosines or adenines within the editing window. Prime editors can make precise insertions, deletions, or all 12 possible base changes. Validation requires checking not just efficiency, but sequence fidelity of the intended edit. Let me give you a real-world example from therapeutic CRISPR development. Companies developing CRISPR therapies test hundreds of guides. They start by designing 200 guides. First filter. Test all 200 in cells and keep the 50 with over 50% on-target efficiency. Second filter. Deep sequencing of 30 predicted off-targets for each, keeping only the 10 guides with less than 5% off-target editing. Final validation in multiple cell types, animal models, and safety studies narrows it to 2 or 3 lead candidates. This rigorous process takes 2 to 3 years and costs millions of dollars, but it's necessary for safety and efficacy. Let me give you quantitative benchmarks to aim for in your own work. A good guide should achieve greater than 60% on-target efficiency with less than 3% editing at the top off-target site. An excellent guide achieves greater than 80% on target with less than 1% off target. For basic research applications, you might accept 40% on target if off targets are below 5%, combined with validation using multiple guides. For therapeutic applications, the bar is much higher. You need greater than 90% on target with less than 0.1% off target editing. Now let me give you practical advice for building your own validation pipeline in your lab. Start simple. Design 3 guides for your target gene. Perform on-target sequencing for all 3. This establishes which guide works best. Add off-target checking for your best 1 or 2 guides. Predict 20 to 30 sites and sequence them. Then validate in your actual biological system of interest, not just in HEK 293 cells. As you gain experience and resources, scale up. Test 5 to 10 guides. Check 50 to 100 off-target sites. Validate across 2 to 3 cell lines. Here are essential computational tools you should use. For guide design, use benchling, CRISPR, or CRISPR-Scan. These incorporate predictive algorithms for efficiency. For off-target prediction, use CASO-F Finder or COSMID. These search the entire genome for potential off-target sites. For sequencing analysis, use CRISPR-Cas2 for NGS data or TIDE for Sanger sequencing. These quantify Indel frequencies automatically. Let me emphasize something important. Negative results are valuable data. If a guide fails to edit efficiently, don't just discard it. Try to understand why. Was it poor transfection? Chromatin inaccessibility? Secondary structure? Failed guides teach you about the limitations and help refine your predictive models. Document everything, even failures. This data is valuable for the scientific community and prevents others from repeating the same mistakes. Let me summarize our 6-step validation workflow for CRISPR editing. Step 1. Define your intended edit precisely, specifying the exact genomic location, guide sequence, PAM, and expected outcome. Step 2. Measure on-target efficiency by deep sequencing, quantifying not just total indels but functional indels. Step 3. Measure editing at predicted off-target sites through targeted amplicon sequencing. Step 4. Compare multiple guide designs systematically to identify the best balance of efficiency and specificity. Step 5. Build predictive models from your accumulated data to improve future guide design. Step 6. Validate your best guides in new biologically relevant cell lines and measure functional phenotypes. This workflow is not optional or excessive. It is the minimum required for rigorous, reproducible CRISPR science. Let me leave you with key principles to remember. First, measure. Don't assume. Never assume your edit worked without sequencing validation. Second, multiple guides provide confidence. Consistent results across guides rule out off-target effects. Third, off-targets must be quantified, not ignored. They can completely invalidate your conclusions. Fourth, cell type specificity matters. Always validate in your relevant biological system. Fifth, build knowledge systematically. Each experiment teaches you something about what makes guides work. CRISPR is an incredibly powerful technology, but power requires responsibility. Rigorous validation is not about being pedantic. It's about ensuring your science is reproducible and your conclusions are correct. The field is moving toward higher standards. Journals increasingly require off-target analysis. Reviewers demand validation with multiple guides. Start building good habits now. Every CRISPR experiment you do should include these validation steps from the beginning. This workflow will make your science stronger, your publications more impactful, and your results more trustworthy. Thank you for your attention to this rigorous scientific lecture on CRISPR validation.